I'm Tim Nelson. I'm the director of the Todd and Karen Wanick Family Program for Hypoplastic Left Heart. Talked to most of you at your poster presentations, and I really enjoyed listening and learning from what you presented. I'm excited for the oral presentations here. So we have some people from the HLHS program. There's some, raise your hand from the program. We got Tim Olson, Karen, Emma in the back there. More people will be coming in. These are going to be your judges, and Tim Olson is the toughest of them all, so keep an eye on him. Honestly, the strength of what I think this session embodies is all of you are now the experts on hypoplastic left heart, which none of you had ever heard of probably a few months back. What I really hope comes out of this next hour and a half, to whatever we're here for, is you guys interacting with each other. And I teed it up for at least one of you that I heard the exact same hypothesis interpreted the different way today. And those of you that I told you probably know who I'm talking about. So you've read the same literature, you've read the same articles, and you may have interpreted that literature differently than your colleagues. And so what I hope that you do today is that you have the confidence to speak up and ask questions. And those of us on the HLHS program can sit and observe and watch. Because what I find the most remarkable is you as the experts will see something that somebody else presents and you'll have a question. And that is very important in the process that you guys are part of right now. It's doing the research, it's putting it together, and now it's being able to articulate that and have a dialogue with your questions and answers. We like to talk in our program, we respect individuals and we challenge ideas. And that's what hopefully is on display here today. We respect all of you, but we challenge every single idea because that's what we do as scientific partners in this journey. So respect each other, but don't hesitate to challenge their ideas, and none of us will be offended. And so I'm really looking forward to some really exciting talks. The way this thing is going to work is you got 10 minutes to present. Everybody knows the rules, right? I get to be the enforcer. I'm Katie's bad cop here. So I'm going to be sitting up front here, and at 9 minutes, I'm going to raise my hand. So you got 1 minute left. And at 11 minutes, I'm going to come up here and drag you off the stage if you're still talking. So you get a 1 minute warning with me raising my hand, and then 11 minutes is the drop dead deadline, and we'll have time 4 to 5 minutes for questions. So be prepared for questions, the rest of you, as you listen to it. So it looks like we have too many Northfield people on this slide if I'm looking at it right. So that's going to be the challenge for next year to the rest of the state to compete with Northfield, it looks like. But nonetheless, let's get started with the first one. That's a 5-minute penalty.
We'll just speed it up. Yeah, we got it.
Ready to roll.
All right. So sometimes a problem can seem a little insurmountable. One such problem was that of surgically repairing fetal hearts affected by hypoplastic left heart syndrome, HLHS. Doctors decided to approach this problem in a unique way. The first surgery was done not to fix every problem seen in HLHS, but rather to set up subsequent surgeries that could treat the entire set of symptoms. We were inspired by their stepwise, innovative procedure and decided that the cause of HLHS might ideologically mirror its surgical treatment. For us, solving what seemed to be an impossibly complex problem began with focusing on valve malformations.
We believe that the strength of our proposal lies in how we connect the significant pathway Notch to the promising yet not well explored pathway of nodal in order to explain HLHS in a comprehensive stepwise manner. We will begin our presentation today with a general overview of the symptoms of HLHS. We will focus on the downregulation of the protein Notch via 3 environmental factors that causes incorrect valvulogenesis. We will next discuss how the cardiac tissue becomes hypoxic under this model due to decreased coronary artery flow. Finally, we will propose that nodal, increased in these hypoxic conditions, interferes with cardiomyocyte maturation, specifically in the left ventricle.
So what we see at the first green arrow is mitral valve malformation. At the second arrow there, we see incorrect aortic valve malformation, which can also impede the progress of blood through the heart. The third arrow will show aortic arch hypoplasia or undergrowth. And the final arrow will display left ventricular hypoplasia, which prevents the left ventricle from functioning.
One important factor controlling proper valve development is the signaling protein Notch. The possibility that the Notch signaling pathway is involved in HLHS is promising because environmental factors such as maternal diabetes, retinoic acid, and the pollutant TCDD downregulate Notch. This downregulation in Notch during cardiac development causes dysplastic AV valves, aortic valve calcification, and aortic and mitral valve regurgitation. To understand how this occurs, we must first understand normal Notch signaling. Signaling begins when specific ligands bind to the Notch1 receptor, shown here. The receptor then undergoes cleavage to form NIC, a transcription factor. One target of normal NIC is to produce a protein called SNAIL. SNAIL decreases the expression of cell adhesion molecules within the endocardium of a normal developing heart. One such cell adhesion molecule is VE cadherin, which plays a vital role in valve development by regulating a process known as endocardial mesenchymal transformation, EMT. During normal EMT, the cardiac cushions, located here, grow from both sides of the heart and fuse, forming a septum and initiating valvulogenesis. This growth occurs when endocardial cells on the inside of the heart delaminate and then differentiate into mesenchymal cells. This delamination requires a decrease in cell adhesion molecules, such as VE cadherin. So, let's pause. Downregulating Notch overexpresses VE cadherin, prevents EMT, and disrupts proper valve development. Therefore, a disruption in the Notch signaling pathway serves as a logical explanation of how valves become malformed in HLHS patients.
It was crucial for us to start with valve malformations when analyzing our systematic stepwise approach to the discovery of the cause of HLHS. However, that's only half the picture. We still haven't described a mechanism by which the left ventricle becomes hypoplastic. To address this, we looked into the literature, and the literature reflected the deep understanding that is presented for how Notch affects valve malformations in a wide array of congenital heart diseases. However, so far, to our knowledge, few theories have presented a full systematic picture of HLHS. So we decided to connect these issues from valve formations to disruption of the left ventricle, and we did this by using the theory of decreased coronary blood flow. Shown here, a malformed mitral valve would cause decreased prograde blood flow from the left atrium to the left ventricle. Additionally, a malformed aortic valve would decrease ventricular output into the aorta. As a result, some blood may come from the left side of the heart, but additional blood will also come up from the right ventricle, up through the aorta, and through the patent ductus arteriosus. This blood would then need to descend the aorta, regurgitating the valves there to reach the coronary arteries, labeled here and here. What this means is that the entire heart would receive a lower perfusion rate of blood and undergo a hypoxic or lack of oxygen condition in the entire myocardial tissue.
Hypoxia upregulates the signal molecule nodal. We believe that in the hypoxic cardiac environment created by Notch downregulation, nodal is upregulated with improper timing, ultimately leading to an underdeveloped left ventricle. Nodal drew our attention as a potential step in the path to HLHS because of its signaling localization. An antagonist called lefty restricts nodal expression to the left side of the heart. Its importance to our model increases upon considering factors of its correct function, which occur during heart tube formation. When properly expressed, nodal directs cell migration to the left side of the forming heart tube. During this process, it has been shown to prevent embryonic stem cell differentiation. Once the heart tube is fully formed, nodal is downregulated and differentiation can resume. In our model, the hypoxia resulting from improper valvulogenesis upregulates nodal after it has been silenced upon completing its role within heart tube formation. Though nodal expression at this later stage of heart development has not been extensively researched, we believe that it would negatively impact cardiomyocyte maturation and differentiation in a manner similar to its maintenance of undifferentiated embryonic stem cells. This localized inhibition of cardiomyocyte maturation would ultimately lead to a hypoplastic left ventricle.
Another five. After reviewing our research on the Notch and nodal pathways, we formulated our hypothesis. We propose that a downregulation of Notch via environmental factors results in malformed mitral and aortic valves, and this causes hypoxia in the cardiac tissue. We propose that subsequent upregulation of nodal due to these hypoxic conditions inhibits left ventricular cardiomyocyte maturation. When looking back at the symptoms of HLHS with which we first started, mitral valve malformations, aortic arch and aortic valve hypoplasia, and left ventricular hypoplasia, our proposal provides a plausible origin for each symptom. Our approach of combining essential pathways does two important things for the field of HLHS research. First, our research highlights the importance of viewing pathways as influencing factors on other pathways. This allows us to see the full physiological scope of problematic development by stepping away from a highly focused look at individual factors. This is done in hopes of providing comprehensive discoveries. Secondly, our approach places a plausible origin in environmental factors that disrupt Notch1 signaling. From our hypothesis, we made two main experimental goals. Our first goal is to illustrate the indirect connection between Notch downregulation and nodal upregulation via the mechanism of valve malformations. We would predict to see a decrease in Notch protein expression and a corresponding increase in nodal expression. Our second experimental goal was to support our idea that increased nodal does lead to left ventricular hypoplasia. We would predict to see a decrease in growth in left ventricular cardiomyocytes. Collectively, we present an underlying cause of hypoplastic left heart syndrome.
We would like to thank Professor Kalyani for her support and guidance, other St. Olaf faculty, and the Mayo Clinic Center for Regenerative Medicine, as well as the Todd and Karen Wanek family Program for HLHS for making this program possible. Thank you for your time.
It's always the hardest to go first, so thanks for them for doing a great job. Questions? And I'm relying on the other teams.
When you talk about the environmental factors that could increase or decrease the Notch, what kind of factors would that consist of?
So, really, the environmental factors led us to choosing Notch itself. When we sat down and looked at this, there's many pathways that we could have chosen. And so the environmental factors that we looked at specifically were reported were TCDD, which is an air pollutant, maternal diabetes, and malregulation of retinoic acid all cause to disruption of Notch. However, there are others, or there could be others that are untested. It was more along the lines of the fact that we saw that these are several different pathways that could possibly lead to this first initiation, and perhaps it's simple gene mutation. But we chose them, and we chose Notch because we found that these were pertinent. And I mean, they all have different pathways too. Specifically, maternal diabetes has a different temporal expression of the ADAM10 gene, which then disrupts Notch. TCDD simply downregulates Notch, and other things like that.
Thank you.
Go ahead.
For the environmental factors that you mentioned, have those been connected to HLHS?
I mean, yeah, some of the reports were actually really interesting to read because sometimes they would connect it specifically to valve malformations, and then that would be connected to a wide array of congenital heart diseases, whereas some specifically said, yes, this is present in Notch, this disrupts Notch, which then leads to cases of HLHS, and other congenital heart diseases, some of them specifically say HLHS. I think it can affect a wide array of things, but HLHS is definitely within there. I think few reports, there certainly are out there, but few reports have specifically said this environmental factor causes HLHS.
You mentioned you had experimental goals as for what your hypothesis was supposed to do, but how would you test that, actually physically tested?
Yeah, we had proposed a few possible experimental setups. For the first goal of linking the downregulation of Notch to the upregulation of nodal, we had thought of using mouse embryos and transfecting those with microRNAs that would first disrupt and decrease Notch production. We could then measure Notch and nodal expression levels with a Western blot test. For connecting the nodal upregulation to the left ventricular hypoplasia, we would use possibly cardiac stem cells, transfect those with mRNAs that would upregulate the nodal, and then we could test the growth of the cardiac myocytes viewing the cardiac troponin markers. That is kind of a little layout of how we do that.
But this is our first experimental goal. This would be our second experimental goal.
Are the Notch and nodal genes specific to just the heart, or are they found in other parts of the body?
They're found in other parts of the body, but because of the timeline of our model and because of, well, the timeline is specific for nodal because it's only being upregulated because of hypoxia that's in the heart already. The Notch pathway, that's actually, the pollutants that we're linking it to have been linked specifically to Notch1 transcription, which then prevents Notch, but that's specific to the cardiac. So yes and no, they are found in more places in the body, but we're proposing a model that only focuses on its expression within the heart.
So on the heart diagrams that you were using, there was no mention of the holes that exist in fetal hearts that allow for the blood to flow between the left and the right sides of the heart. I was wondering what you guys, how you guys addressed that, because you talked a lot about how the blood flows, and I don't think that was mentioned.
Yeah, sure. So the two main holes I think you're referring to would be the patent ductal. Oh my God. Sorry. Sorry. I thought everything was going wrong. But anyways. So I think what you're referring to would be the patent ductus arteriosus and then the connection between the atria. So that's really part of our introduction speech, which was the surgical treatment. So what's done with that is keeping the patent ductus arteriosus open so that the right ventricle can provide both systemic and pulmonary blood flows. But
I can add on to that if you
yeah.
want. Yeah, oh, sorry, man. If you also, I mean, like, if you think about it, like, what really is the deal is that, like, there's a decreased pressure, you know, and so the decreased perfusion rate of the blood is what is causing hypoxia within the entire myocardial tissue. Does that kind of answer your question, or are you still wondering about that?
Somewhat, it's...
Okay.
Any other question? Yes, sir.
I think you talked about using the microRNA to disrupt the Notch signaling genes. How exactly would you want to disrupt it? Is it just like you want to delete the segments, or do you want to mutate it? How, and then what kind of like effect would you expect to see?
Yeah, microRNA, it's a complementary base pair sequencing that will attach to the mRNA strands existing already, and depending on how complementary it is, it will either destroy the mRNA sequence or it will just prevent translation of it. We hadn't really gone into much further detail of how we would exactly plan that out, but what we would predict to see would be after disrupting the Notch production, we would then measure and verify those decreased levels of Notch, and then in later development test for the levels of nodal and see the upregulation of nodal because of the hypoxia induced by the initial downregulation of Notch.
My name is Abhishek Chandra.
I'm Jack Goldstein.
And I'm Daniel Hogan. For our project, we explored a possible interaction between inflammation and HDAC expression as a possible cause to hypoplastic left heart syndrome. HLHS is a congenital heart defect that presents with a malformed left ventricle. It may also present with a malformed aorta, an open ductus arteriosus, and an atrial septal defect. HLHS is a life-threatening condition that occurs in 2 to 3 out of every 10,000 births in the United States. And currently there is no known cause to HLHS, but there have been numerous studies into a number of different genes and environmental causes. And while correlations have been found, there's no causal relationship that has been identified yet. So we hypothesize that an overexpression of HDAC3, which is triggered by a source of severe inflammation, will reduce the expression of NKX2-5, a gene crucial in cardiogenesis, resulting in HLHS. So this is our proposed inflammation pathway. So any source of inflammation will cause the thrombospontin 1 levels, or thrombospontin 1 concentrations to rise. Once this TSP1 concentration has risen, it will bind to the CD47 receptor, which then activates the CA-MK2 channel. Once this channel is activated, it will increase the concentration of HDACs, resulting in a reduced expression for the affected genes. So that last point about how the HDACs cause a decreased expression is very important to understanding our proposition. So in its natural state, DNA is heavily methylated. And this methylation is a process by which methyl groups are added to DNA segments, which causes them to wrap tighter around histone proteins, which restricts gene transcription. On the other hand, histone acetylation is a process by which acetyl groups are added to histone proteins, unwinding this DNA and enhancing the effect of gene transcription. So HDACs are enzymes that will remove these acetyl groups from the histone proteins, mimicking the effect of methylation by rewinding the DNA around the histone cores and reducing the expression of affected genes.
So of the number of genes that have been studied as possible links or relationships to HLHS, we wanted to focus on one in particular, which was NKX2-5. This gene has been shown to have a direct link to left ventricle formation. It also works through the expression of the BMP2 protein and the HAND1 gene. This gene has been shown to have a reduced expression in patients with HLHS, which is just additional information to help support our proposal. The most important thing from this slide, though, is that it's regulated heavily by the canonical Wnt pathway. And so what this pathway does, basically from a simple level, is it just controls when the gene is expressed and to what extent that occurs. So in its off position, I'm going to go through this kind of quickly, we can go back into more detail later. Beta-catenin, which is an enhancer, is unable to bind to promoter sequences when the Wnt pathway is turned off. That green DSH is a disheveled protein that destroys the destruction complex beta-catenin, and this prevents target genes from being expressed. So we see degradation, and as a result, we see genes aren't being expressed. When the Wnt pathway is activated, we do see the Wnt protein binding to the frizzled complex on the plasma membrane. This allows beta-catenin to bind to the actin complex in the cytoplasm without being destroyed by the disheveled protein. This results in its ability to move into the nucleus, bind to transcription factors that are on the promoter sequence, resulting in the activation of Wnt target genes. So the Wnt pathway is very interesting. It might make sense to say that there's a mutation in the pathway itself that's causing this reduced expression. However, it's a highly conserved pathway that exists in both plants and animals, and it's in control of a number of genes. And for that reason, we didn't want to suggest that there was a mutation here because it would likely affect other gene expression, likely resulting in death rather than this specific defect. Also importantly is that for the pathway to work in its entirety, the DNA sequence of the gene in question has to be acetylated. It can't be wound around those histone proteins and needs to be available for transcription factors to bind to it. So what we were proposing, or the crux of our proposal, is that we're interrupting this pathway's normal functioning. By an overexpression of HDACs, we can interrupt the pathway by rewinding DNA around histone cores, preventing transcription factors from binding. In our case, this would result in a decreased expression of the NKX25 gene. So our root cause for this was inflammation, and that's a really big topic, and so we need to set up some parameters to help understand what that means for our proposal. So firstly, our inflammation for this proposal has to be really severe and is more likely chronic than acute. For the cascade inflammation pathway that we suggested, thrombosponin 1 levels need to be increased to a point where CD47 receptors are saturated, resulting in an overexpression of HDAC rather than what is normally produced. And this type of inflammation effect is more likely chronic than acute. Because ventricle formation happens really early in a specific time frame during pregnancy, it's more likely that there's an eventual shift in equilibrium as a result of inflammation or a stressor compared to a random separate event that just so happens to affect the fetus at a certain time point. And so that brings into the issue this specific time of effect. It's a really small window in terms of days, but it's really specific when it comes to cardiogenesis, day 15 to 17 of pregnancy.
So this raises one of the biggest issues with our proposal. How would the mother be able to tell that something is wrong? Well, unfortunately, this is currently not possible. Since we are proposing that the damage to this pathway is occurring so early on in the pregnancy, between 15 and 17 days after conception, it is very unlikely that the mother would even know she is pregnant. Additionally, the presence of inflammation may be asymptomatic. Inflammation comes from many different factors, including autoimmune diseases, which often can be asymptomatic and can go undiagnosed. With the combination of how early the damage to this pathway is occurring and the possibility of inflammation being asymptomatic, it is currently impossible to determine the presence of this disease until later stages of pregnancy. Furthermore, the presence of inflammation may have a partially lasting effect on the fetus. This coincides with the findings that other cardiac malformations are also present with HLHS. Here you can see a picture of our proposed interaction pathway. You can see how the inflammation interrupts the normal functioning of the Wnt signaling pathway. The overexpression of HDACs act as a roadblock for transcription factors and their ability to transcribe genes. This eventually leads to a reduced expression of the gene NKX25 and ultimately HLHS. Furthermore, exploring the possible cause incorporates the topic of nature versus nurture. Our proposal investigates the interaction between a namely environmental factor in inflammation and the fetus's genetic predisposition to HLHS. Our proposal is innovative in that it suggests a new paradigm of research in this field. A combination of wet lab studies and epidemiological research would be very helpful to better our understanding of these cardiac malformations. It would be important to determine if any correlation between a stressful environment during pregnancy and the presence of HLHS exists through epidemiological research. The environmental effects on cardiogenesis have been well studied and well reported in other cardiac defects. This is why we determined this would be a good place to start our research. In this way, by exploring inflammation and its interaction with molecular pathways, we are bridging the gap between nature versus nurture in terms of understanding developmental diseases such as HLHS. The implications of this proposal and approach are widespread. Through transgenic animal trials, stem cell research, and epidemiological studies, we can gain a better understanding of cardiac malformation. Hopefully we can determine the root cause of hypoplastic left heart syndrome. This proposal has important implications for both the care and early diagnosis of HLHS and possibly for the prevention of it in the future.
We'd like to extend our gratitude to Dr. Everson, who is our advisor. Though he could not be here today, he provided a lot of assistance for us, to Dr. Shirky's son, and Dr. Freeling for their scientific consult, to the rest of the St. Olaf Science Department for all the help that they gave us, and to the leaders of the Impact Program for the ability and opportunity to participate. I'd like to open it up for questions.
So there's a large number of disorders that could lead to chronic inflammation. Has there been any connection to such disorders in HLHS?
From the research that we were comparing this to, it was related to Holt-Oram syndrome, and it was another cardiac defect related to the gene TBX5. And the inflammation that they were studying was based more off on history, and so pregnant mothers who were in abusive relationships, in war crime situations, and more environmental causes. There have been some studies to suggest that there are conditions such as autoimmune disorders, though nothing has been completely conclusive, though they are finding stronger and stronger correlations as they do more research.
Why is NKX25 the only gene that's affected by the inflammation?
So Daniel spoke on this, sorry, Jack spoke on this a little bit earlier. You can go back to that slide. And we talked about how inflammation can have a lasting effect. And so because NKX25 regulates other gene expressions as well, it sort of serves as an umbrella that does impact other genes along associated with it. And that's what we see when we see HLHS patients sometimes presenting with other malformations. For example, not every HLHS patient will present with all the mutations that we have been listed here. Some will and some won't, and those are controlled by the expression of other proteins and genes, controlled mainly through NKX25. There have been other genes studied, but this seemed most promising based off some of the literature that we were reviewing.
You mentioned that support of your hypothesis could lead to the prevention of HLHS. How exactly could you prevent this?
We were thinking of maybe finding a way to determine the presence of this inflammation prior to that 15 to 17 day range. If there was a way that we could come up with a test to determine that inflammation is present, we could maybe find a cure for this beforehand.
That might be a job for regenerative medicine using stem cell research. Hopefully as it advances, if this all correlates and we do find a cause, utilizing pluripotent stem cells to help fix the heart before it gets too bad might be later.
Is there any way this would need to progress to maybe just education to a mother who would, is not necessarily pregnant yet, but is trying to? Because 15 to 17 days is so early, a lot of women don't know they're pregnant. You know, how could they stop doing something if they don't even know that they're pregnant?
So the inflammation we were talking about, yes, you can be stressing yourself out, you can induce your own inflammation, maybe not intentionally. Sometimes when it comes to that type of advice for a pregnant mother, it would be for someone who's thinking about getting pregnant. The basic advice is trying to avoid stressful situations. Some of the other groups mentioned possible stressors, things that increase cytosol levels, for example, avoiding toxins and other such pathogens are one way to decrease stress and any pathogens affecting the body. But some of the situations that we were talking about and other research was pointing to in terms of cardiac defects was abusive relationships and other really more environmental stressors that were kind of hard to control biologically.
Questions? Can I ask a quick question? I'm not sure if I understood this correctly, but you said there was probable chronic inflammation, but then the problem occurred between days 15 and 17. How would you reconcile those two aspects?
We were proposing chronic inflammation because it would happen in this very specific time, and that it would be less likely for a source of acute inflammation to happen between the 15 and 17 day marker. So if there was a prolonged period of chronic inflammation while day 15 to 17 passed, that's kind of what we were proposing.
As a tag off of that, I guess kind of what we were thinking about is how
and maybe you said this and I just missed it, how is this selective then to cardiogenesis and cardiac development, whereas inflammation, if that's present and is affecting genes, couldn't affect other forms of development? I mean, this is so early right after registration.
So, like you said before, inflammation does affect other mutations. Yes, they are related to the heart. The body has a way of recalibrating once you have an overexpression to HDAC. So while the body is also fixing itself, this equilibrium that we were talking about before does reverse, and the body will fix itself. Inflammation has been known to cause other defects and other conditions, and we have seen that some HLHS patients do present with other conditions, some of which you mentioned were
Trisomy 13 and 18 were common.
And so we're suggesting it does have a long-lasting effect. The more specific part of it is that it needs to affect by day 15 or 17 of pregnancy.
Great, thank you.
Good afternoon. We are the MSU Moorhead Impact Group presenting on the role of homocysteine regulation and microRNA in hypoplastic left heart syndrome. My name is Jake. I'll be giving a quick overview of our presentation. I'll be doing the introduction. Vincent will be covering the hypothesis as well as the rationale. Benin will be covering the innovation that our research is offering, and Gabe will be going over the significance. Hypoplastic left heart syndrome, or HLHS, affects right around 960 newborns every year in the United States alone, with a fatality rate of about 25%, reduced life expectancy, and a relatively reduced quality of life due to a multitude of deficiencies. It is very important to find the underlying cause of this condition. We set out to do this. Our research led us to a study involving pregnant women carrying fetuses with congenital heart defects. In this study, they found a correlation between the upregulation of the microRNA, HSA microRNA 99A, and congenital heart defects. In addition, several studies also found a correlation between the elevated level of homocysteine and cardiovascular disease. We then looked at the biochemical function of homocysteine. We found that it's a precursor to two essential amino acids, methionine and cysteine. It then became our objective to establish a connection between homocysteine, microRNA 99A, and fetal heart development.
In light of the information presented by Jake, we hypothesized that the upregulation of the microRNA, HSA, MIR99A, will lead to an accumulation of homocysteine by interfering with the production of the enzymes methylene tetrahydrofolate reductase, or MTHFR, and cystathionine beta synthase, or CBS. The shortage of these amino acids between weeks 3 to 9 of fetal development causes hypoplastic left heart syndrome. To provide a rationale, let's start with microRNA. What is a general consensus that microRNA can render MTHFR and CBS deficient. But these are key enzymes in the conversion of homocysteine to cysteine and methionine, which are key to protein stability and function. To dig deeper, let's take a look at the methylation cycle, or the biochemical pathway involved in the with homocysteine. Folate is a water-soluble B vitamin that converts. Folate is a water-soluble B vitamin, and the folate cycle consists of a series of reactions that convert folate to methionine. And the enzyme MTHFR plays a key role in that process. Once methionine is formed, it goes through the methylation cycle and is converted to homocysteine. And then homocysteine proceeds through the cycle and is converted back to methionine, and the process repeats all over again. This is necessary to maintain sufficient level of methionine in the cell. And when enough methionine is produced, homocysteine is converted to cysteine instead. And the enzyme CBS catalyzes this reaction. This figure shows normal production of methionine and cysteine and functional MTHFR and CBS. We propose that our microRNA in question binds to the messenger RNAs responsible for the translation of these two enzymes, and a downstream effect of that will be an accumulation of homocysteine and a reduced production of cysteine and methionine, which ultimately play a key role in protein stability and function. Especially, methionine and cysteine play an important role in the hydrophobic effect, which is a major determinant of native protein structure. Additionally, cysteine is the only amino acid capable of making disulfide bonds. And disulfide bonds are the strongest bonds that hold proteins in their various shapes. The fact that methionine and cysteine are the least common amino acids in protein amplifies any effect in the deficiency. So it is reasonable to hypothesize that a deficiency of these key enzymes within very critical periods, weeks 3 to 9, of fetal development will lead to hypoplastic left heart syndrome.
Now, if our hypothesis is proved to be valid, this could have a large effect on the healthcare field. MicroRNA research is relatively new, and there still needs to be more found out about how microRNA interacts with messenger RNA, and then how that later affects the production of enzymes and proteins. For our purposes, we'd like to see how our microRNA HSA99A could be used as a possible biomarker for HLHS. To the best of our knowledge, we found that our hypothesis is the first attempt to make a relationship between HSA99A and the accumulation of homocysteine, which then causes HLHS. If it could be found that HSA99A could be used as a biomarker, there are many applications from this. Many other microRNA molecules are already used as biomarkers in other diseases and conditions. And microRNA is actually a really good molecule to be used as a biomarker for many reasons. It's tissue specific. It is relatively stable in the environment that it's found in. It can be measured and compared in, for our purposes, blood plasma. And it is also relatively well conserved between different species. We would like to use the mouse model to be able to test, at a comparable stage of development to the human 3 to 9 weeks. And we'd also like to use a human cardiac cell line to see if there was an upregulation of our specific microRNA, how that would affect the levels of homocysteine in those cells. We'd also like to propose using non-invasive type methods, such as measuring the microRNA levels in the peripheral bloodstream of pregnant mothers.
The use of non-invasive prenatal testing can lead to early detection of HLHS without the risk of harming the fetus. Early detection will effectively narrow the focus and lead to more specific research. This will significantly increase the likelihood of pinpointing the general underlying risk factors and the mechanism at play. A narrow time frame for etiology will also enable researchers and scientists to focus on that mechanism and from there develop curative therapies. Cell therapy and other regenerative medicine techniques are already being implemented in clinical studies. Additionally, a biorepository has been established for current and past HLHS patients. This biorepository consists of paternal and maternal DNA, as well as the patient's DNA, environmental factors, diet, and any other underlying conditions that can contribute to this disease. It is then reasonable to test our hypothesis by using comparative analysis of the biorepository and examining the microRNA expression and the homocysteine levels. In closing, we propose that that microRNA interferes with the production of MTHFR and CBS enzymes. Subsequently, homocysteine cannot convert to cysteine nor be remethylated to methionine. Both of these amino acids play a role in the hydrophobic effect, which is the major determinant of native protein structure. In addition, cysteine is the only amino acid that can form disulfide bonds. This is the only covalent interaction in protein stability and is the strongest individual bond. With this information, we then propose that insufficient levels of methionine and cysteine can lead to improper protein structure, stability, and folding. We believe that non-invasive prenatal testing to confirm microRNA and homocysteine as disease factors will lead to breakthroughs in regenerative medicine techniques and genetic screening. Narrowing the focus and implementing the use of these biomarkers can lead to prevention, or better yet, a cure for hypoplastic left heart syndrome. Thank you.
Go ahead. So, as you addressed, cysteine is a very vital component in protein structure. So, in many different proteins. So, what other proteins do you think a lacking cysteine? If you're lacking in cysteine, what other proteins do you think would be affected? And could those other proteins being affected produce symptoms that would come with HLHS? Or what would you expect?
Okay, so if I understand your question right, you're asking is cysteine, what protein specifically cysteine could be involved in, and how that can lead to HLHS?
And what symptoms would you see alongside HLHS? Perhaps not related to the heart, but in keratin structure or whatnot.
As far as cysteine and methionine go, they're essential amino acids that are involved in a multitude of proteins. We didn't pinpoint specific proteins that they are involved in as far as the left ventricle formation, but they are rare amino acids, relatively speaking. So the amplified effect of that is if they're already rare, then they could affect the protein stability because if they're insufficient levels and they're already not there. But for specific proteins, I'm not sure.
I think this is kind of going off of what he was asking. But how is the loss of these very vital amino acids, how is this specific to HLHS? And if a developing fetus is lacking in these amino acids, wouldn't we expect to see far more ranging developmental problems?
So specifically to HLHS, cysteine and methionine, we are proposing that methionine, which is a general consensus, is involved in protein folding and coordinating that, and cysteine with the stability. We're proposing that this renders the proteins weak. That does not necessarily destroy the protein, but any change in temperature, pH, or environmental factors inside in utero can affect the folding from day 21 to day 28 and lead to a malformation in the ventricle formation. As far as throughout the body, this microRNA that we've established or that we've correlated is involved in cardiogenesis, and that's why we picked this one. So there could be other effects of the shortage of cysteine in any proteins in the body, but we focused in on this one because of the established connection with the cardiogenesis.
Yes? I have a question about the non-invasive prenatal testing that you were referring to.
Yes.
What kind of testing and when were you looking
Okay,
at and when?
so this is very important, yes. What we can do for biomarker testing, it's a blood draw. So we can do this as early as we want to in a pregnancy, and with the changing in healthcare and if the woman was in there early enough, but we can take a blood test and analyze a multitude of microRNA expression very easily and accurately, and it doesn't harm any, it's not an amniocentesis or anything invasive, so it is a blood test, yeah.
So, essentially, like, would you only be doing this on mothers of HLH that already know they have HLHS children, or like before they even have their anatomical ultrasound?
So that's where the change in healthcare would have to come around. So it would have to start with a developed genotype that may be a risk factor, or any other risk factors as far as this is a hereditable disease, so if you're at risk genetically. It would start with them, and then as microRNA and homocysteine are developed and confirmed as biomarkers, it could cover a broad range of pregnancies from there. But they would have to be in early enough, and that does not happen often enough.
Yes.
So is this microRNA usually expressed during cardiac development, or is it abnormally expressed in cardiac diseases?
It's just overexpressed, really, and specifically with cardiogenesis and congenital heart defects.
Do you have any idea what might cause those changes in expression?
As far as overexpression of the microRNA, we did look into that, and there's so much study on microRNA and how it can work and the different functions it has, but what we did come across was reactive oxygen species can possibly misregulate or dysregulate, causing overexpression and underexpression from oxidative stress. There wasn't anything conclusive on that, and that's where the research would have to lead from here.
One more question?
What kind of, let's say you do a blood draw and you determine that there is a problem. What sort of prenatal care can you think of to give for the mother to prevent any further, abnormal heart development?
So what we can do, and what some studies have talked about, is when you have a shortage of methionine, you can supplement with folate, and it goes through a folate cycle and can produce methionine. The problem with that is if you still have a deficient or shortage of cystathionine beta synthase, you still can't get homocysteine to convert to cysteine. So even if you can get it to remethylate to methionine, once the body knows that that is sufficient, it's going to tell you to stop making it. So the curative therapies, if we identify this as a mechanism, we would have to do a human cardiac cell line or anything like that to experiment with what we could do to kind of bypass and get homocysteine out of there.
Great, thank you.
So we're the group from Carleton College. I'm Madeline, this is Sandy, and this is Haley. And today we're going to talk about how FHL2 can modify the activity of beta-catenin to decrease cell proliferation in the left heart. So a little bit about heart development. There's one progenitor cell type for the heart that develops into a heart tube by around day 21 with an inflow and outflow tract. And we're mostly interested in the left ventricle, which is labeled LV and is light yellow in all three figures. During normal heart development, there's accelerated cell division on the left side of the heart, which leads to heart looping, which you can see by around day 28. And this looping changes the hemodynamics of the heart, or the way the blood is pumped, so that when looping occurs, the blood begins to be pumped more efficiently and in a wave-like pattern. So what can cause changes in heart development? Changes in gene expression can affect cell proliferation, which affects heart development. For example, accelerated cell division on the left side of the heart during normal development is important for left-right patterning. Changes in hemodynamics, or changes in pressure and stress, have been shown to cause changes in gene expression in certain cases, and so then that goes back to point 1 and can also cause changes in heart development. So our hypothesis is that FHL2 is modified, and that affects the way it interacts with beta-catenin, causing decreased cell proliferation, leading to HLHS.
So why do we choose FHL2? Well, first of all, we know that FHL2 is only expressed on the left side of the heart. This makes it an excellent target gene to look at because it only affects the left side development and not the right side development. We also found that FHL2 is involved in the control of cell proliferation and cell differentiation, which is something that we were interested in due to left-right patterning. We know that FHL2 binds to transcription factors to either enhance or inhibit their activity, and in cardiac cells, we know that FHL2 binds to beta-catenin. Now, what is the significance of that? Well, beta-catenin is a part of the Wnt canonical pathway, and when it's released from inhibition, beta-catenin can enter the nucleus, bind to an enhancer region, and upregulate the transcription of target genes involved in cell proliferation. Now, we're proposing that FHL2 is binding to beta-catenin, and we know that this happens because FHL2 can enter the nucleus and interact with beta-catenin once it's already bound to its enhancer region. So we believe that FHL2 is binding to beta-catenin and inhibiting its ability to upregulate target genes, and this causes a decrease in cell proliferation. So what's different about HLHS hearts as compared to normal hearts? Well, the diagram on the left shows a picture of an HLHS heart, and the diagram on the right shows a picture of a normal heart. And as you can see here,
the left ventricle of the HLHS heart is underdeveloped as compared to the left ventricle of the normal heart. So left side structures are underdeveloped in HLHS. However, there are also a few key differences as well. Inhibitors of the cell cycle are upregulated during HLHS, and there is decreased cardiomyocyte proliferation. So here's our proposed gene expression pathway. We believe that SRF and NKX25 upregulate FHL2. FHL2 then binds to beta-catenin and decreases cell proliferation, changing the development of the left ventricle. So in HLHS, we see an overexpression of FHL2, which causes FHL2 to bind more frequently to beta-catenin, causing decreased cell proliferation and a smaller left ventricle that's underdeveloped. Okay, so in a previous slide, we compared the HLHS heart with the normal heart. And on this slide, we've also added the hypertrophic cardiomyopathy heart. And throughout our research in HLHS, we came across hypertrophic cardiomyopathy, and we found a paper by Tran et al. In 2016 that linked hypertrophic cardiomyopathy with FHL2. And so FHL2 is known to directly affect the development of the left ventricle, which they showed in this paper. And they've also already established a link to FHL2, which we're hypothesizing that changes in the FHL2 expression can create a spectrum of phenotypes, where in hypertrophic cardiomyopathy, it's underexpressed, and in HLHS, it's overexpressed, leading to the changes in the left ventricle. And then FHL2 has also been linked to cell proliferation, and we're hypothesizing that a decreased cell proliferation and differentiation could cause this stunted growth in the left ventricle. So here's the pathway that we showed before, where we've added in the hypertrophic cardiomyopathy side. So to compare the two, FHL2 in hypertrophic cardiomyopathy is underexpressed, meaning it isn't present as much in the cell to bind to beta-catenin, leading to more cell proliferation and an increased left ventricle that's overdeveloped. Where we're hypothesizing in HLHS hearts, FHL2 is overexpressed, meaning there's more in the cell to bind to beta-catenin, leading to less cell proliferation and a decreased left ventricle that's underdeveloped. So we came up with some ways of testing the role of FHL2 in HLHS. The first thing that we would want to establish in our research is to know whether FHL2 is actually upregulated and more present in HLHS cells. So we could do this by looking at a Western blot. The second question we want to ask is, would changes in FHL2 in model systems cause HLHS-like phenotypes? And you could use this by doing CRISPR-Cas or fluorescent marked assays of cell proliferation. And the last question would be, if you can upregulate it and cause the HLHS-like phenotypes, can you also use siRNA to knock down the expression of the gene and revert it back to normal phenotypes? So the significance of the FHL2 role in HLHS is that you could do a genetic screen to identify those who are more likely to develop HLHS. And this could be done during in vitro fertilization and during the embryonic stage. And you could select against embryos that are more likely to develop HLHS. And down in the future, there's also a potential of correcting the gene using CRISPR-Cas9 or viral vectors. And then we'd like to thank Mayo Clinic and Katie Campbell for the program, and Carleton College and Jennifer Wolf for help along the way. Yeah?
So you mentioned that the factor you're going after, FLH2, is limited to the left side of the heart.
Mm-hmm.
Why is that not seen in the left atrium?
Well, it's seen in other left side structures, not just the left ventricle.
Okay.
Yes?
So off of his question, how come we then also see mutations in the aorta and the septal defect and not just the left ventricle with the LH2 that you closed?
That goes along with our feedback loop where the left ventricle starts to be underdeveloped. So the blood starts pumping through the right side more and the hemodynamics change so that it becomes like a bad feedback loop where it just gets worse and worse over time. Yep.
Going off of that now, so if the bad feedback loop restricts it to the left ventricle, how come the right side isn't hypoplastic?
Oh, well, so what restricts it to the left side is FHL2 expression.
Right.
So you're changing the hemodynamics so that blood gets shunted to the right side. And we've actually seen papers that show, due to the change in hemodynamics, there's restructuring of the right ventricle, and it begins to look more like a normal left ventricle. Yeah?
So you said that FHL2 binds to the beta-catenin and it restricts some genes, but what type of genes would you be referring to? I know it wasn't on your diagram, but what kind of genes would reduce cell proliferation?
We haven't currently looked at any genes in particular, but we saw a study by Regenerative et al. In 2013 that demonstrated that this binding, overexpression of FHL2 binding to beta-catenin changed cell proliferation or cardiomyocyte proliferation. Yes?
As far as treatment or significance, you guys talked about genetic screenings. Is there any other options for it? Like if they were found to have this FHL2, what else could you do besides just not having a kid? Or
Well, we mentioned some genetic therapies that you could do, so potentially you could correct the sequence with CRISPR-Cas in the future. I guess another thing that potentially could be done in the future, which I don't know if much study has been done into, is doing siRNA, so knocking down the overexpression of the protein could potentially help make it normal.
Other questions? So you said something about the, see now when people don't ask questions, then I get to ask questions. So, because, I got 2 minutes for questions now. You said in vitro fertilization and genetic testing there. Tell me more of what you're thinking about that. Not all babies are conceived with in vitro fertilization. Would this just be limited to in vitro fertilization from a diagnostic perspective?
Potentially you could also use it if the parent has HLHS and potentially test to see if they have a change in their genetic
Or if
sequence.
they've had children that have had HLHS in the past.
So does this get added to Facebook then, your genetic makeup before you pick your partners? Pick and choose wisely. It's a fascinating debate because there is actually an increase. There seems to be an association that we're interested in with in vitro fertilized babies that have an increased predilection to some of these things. So there is potentially a way that would be clinically relevant to be doing that with an in vitro fertilized type of platform. So it's just interesting to get your thoughts with it. Any other questions, challenges, difference of views? We're challenging ideas, respecting each other, remember?
Yeah.
One more?
You were just saying that if it's knowing if they have HLHS in the family, did you find research that there was connection or links between, because from what I know of HLHS, there's very little link within families having multiple cases of HLHS.
I don't think we found any research that specifically said that if you have someone in your family that has HLHS, you're more likely to have children who have HLHS. But I think those might be the people who would seek out the genetic testing.
Okay.
But you know, the beauty of today is that we got Tim Olson sitting here, who's the world's expert on that. Can you answer that question?
Yeah, so it's very, very rare to see 2 children in the same family with hypoplastic left heart syndrome. We've identified such cases through our registry, our research study. More commonly, though, is to see more subtle congenital heart defects within families. So there's a paper that came from our group recently that described increased frequencies of congenital heart defects you may be asymptomatic from. For example, a bicuspid aortic valve. The aortic valve, as you know, has 3 leaflets. Some family members have been found to have 2 leaflets, and other such congenital heart defects. So there does seem to be genetic background in certain families if you do screening echocardiograms on parents and siblings.
Yeah, and that connects kind of a little bit to our hypothesis where we believe there's a spectrum of phenotypes. So one child might be more severe than the other child.
Great, thank you.
Hello, we're from St. Olaf. My name is Leah Andrews.
My name is Thomas Olson.
I'm Nita Rupitamahania.
I'm Pete Smith.
And I'm Andrew Salee, and we'll be telling you why, perhaps, the blocking of cardiac gap junctions may trigger HLHS. So, as has been previously discussed, HLHS, or hypoplastic left heart syndrome, is the underdevelopment of the left side of the heart in fetuses. And while it's rare, it's quite common for a congenital heart defect and requires invasive surgery in order to be acted upon. Even with this surgery, however, survival rates are incredibly low. And to top this off, we don't yet know the cause to HLHS, which is the whole point of the IMPACT program. So when considering what might cause HLHS, we have two pathways in which we can look at things. We have a genetic aspect and an environmental aspect. And when we look at the genetic aspect, we see that there are racial splits in prevalence, that there might be some heredity aspect, as has been previously discussed. When we look at the environmental aspect, we see that perhaps different things in the environment may cause HLHS. We see a seasonal trend with HLHS increasing in prevalence during the summer and decreasing during the winter. And we even saw an outbreak of HLHS in the 1980s in Baltimore, where every single section of this indicates an outbreak, a case of HLHS, which is, as we can see, this is localized to a very small area of Baltimore. So this seems almost like an infectious outbreak occurring of HLHS, which is bizarre, considering that it is a congenital heart defect. So we wanted to consider both the genetic and the environmental aspects of HLHS when making our hypothesis.
Considering the two significant trends as noted by Andrew in the previous slide, we hypothesized that HLHS is a result of the disruption of cardiomyocyte gap junctions due to either connexin 43 mutations or autoimmune damage induced by anti-streptococcal and protein antibodies. Our hypothesis is unique in that we are proposing two routes to HLHS, one being genetic and the other being environmental, both resulting in the disruption of cardiomyocyte gap junctions and thus leading to HLHS. Gap junctions are organized collections of protein channels in cell membranes that allow ions and small molecules to pass through. Gap junctions are particularly important in cardiac muscle because the signal for the heart to contract is passed efficiently through these gap junctions. The proteins that make up gap junctions are comprised of units called connexons, in which two connexons here make up one gap junction. Those connexons are comprised of further subunits called connexins, in which 6 connexins make up one connexon. Shown here is the carboxyl terminal of one connexin. This structure is extremely important in the assembly of gap junctions. Connexin 43, in particular, is significant to our hypothesis in that it allows for gap junction intercellular communication between cells and is an important protein in heart gap junctions. The diagram here shows the phosphorylation sites of connexin 43. These sites are extremely important in that they are critical in managing the life cycle of connexons. The life cycle of connexons begins with the synthesis of connexins, and without this synthesis occurring, for example, because of immunoglobulin antibodies, the life cycle of connexons is thus affected.
So going down the genetic route, we have connexin 43. Now, connexin 43 is a protein in gap junctions that is encoded by the GJA1 gene. Now, a mutation to the GJA1 gene would prevent phosphorylation, thus disrupting the life cycle of the connexon. In a study done in 2001, the mutated GJA1 gene was observed in almost 80% of its HLHS subjects, but in all of the controls who had no HLHS, there was no mutation observed whatsoever. In the same study, it was also observed that there was a 97% nucleotide similarity
between the mutated GJA1 gene on one chromosome and its pseudogene on another. This would suggest that there was an illicit recombination of the two chromosomes, and this recombination may lead to a mutation, and we would expect that mutation to disrupt the gap junction function, ultimately leading to HLHS.
While the connexin 43 mutation may be a potential route to HLHS, it doesn't explain why HLHS increases in prevalence during the summer and occurs in sporadic outbreaks throughout the years. So with further research, we have found that the bacteria, Streptococcus pyogenes, which causes the disease strep throat, has been linked with HLHS. So strep throat is a contagious disease that peaks through the late fall through the early spring, and which can explain HLHS's increased prevalence in the summer because HLHS develops between the first and second trimester of gestation. Now, since strep throat is also contagious, outbreaks in strep throat could lead to outbreaks in HLHS. Now, when a person is exposed to the bacteria Streptococcus pyogenes, their body produces antibodies in response to specific antigens. So the Streptococcus M protein antigen, antigen invokes the body to produce these anti-strep antibodies, which actually have been known to cause autoimmune damage and have led to diseases such as rheumatic fever and rheumatic heart disease. In addition, these antibodies are immunoglobulin G, or class IgG antibodies, and they have the ability to cross the placenta. So because of this, investigators have taken these antibodies and injected them into rats, which have then been impregnated and have produced offspring that have had the HLHS phenotype. So because of this, it's implying that something about these maternal antibodies is causing damage to the fetus. Now, with further research, we have found that the class of IgG antibodies is actually able to block the phosphorylation sites on that carboxyl tail of the connexin 43 protein, which we had discussed earlier. By blocking these phosphorylation sites, it would prevent phosphorylation, which would affect the connexon life cycle and disrupt the normal regulation of the gap junctions, leading to HLHS. So therefore, we have found two independent routes that lead to HLHS because they both disrupt the normal function of gap junctions. We have found our genetic route, which has been involving this GJA1 mutation, which prevents phosphorylation and leads to the misregulation of gap junctions. And we have found our environmental route, which is due to the antibodies that are produced in response to the Streptococcus pyogenes bacteria, which have caused an autoimmune reaction with our carboxyl tail, which prevents phosphorylation and also leads to the misregulation of gap junctions. And since gap junctions are crucial to the cell proliferation and cardiogenesis, we're proposing that it would then cause HLHS.
So in order to test both of the different factors which affect HLHS, we proposed to first explicitly test whether these things are affecting the gap junctions. As sort of a proof of concept, we would use established methods of fluorescent microscopy to observe whether these, either the mutation or the anti-strep antibodies impair the spreading of these fluorescent dyes, such as what you can see in these. Once that proves to be true, we would move on to a more macro experiment. Recent paper published in Nature outlines a process which uses the scaffolding of a decellularized mouse heart and human-induced pluripotent stem cells in order to recreate a miniature human-like heart with human cells. This is important for our testing because this heart has the ability to exert mechanical force as well as respond to drugs. Also, this heart is as the heart is growing, the stem cells are able to proliferate, differentiate, and migrate into their specific tissues with the introduction of growth factors. So our deviation from this second experiment would be in experiment A to introduce these IgG anti-strep antibodies at the same time as the perfusion of growth factors. And in the other test, we would intentionally mutate the GJA1 gene using CRISPR-Cas9 before the recellularization process begins. in both of these experiments, in comparing them to a control heart, we would expect these hearts to exhibit HLHS-like symptoms, such as the malformity in the left ventricle. So looking forward, if our hypothesis proves to be true, we expect these following treatment options to reduce the prevalence of the disease. With respect to the environmental cause, it's fairly straightforward in that the diagnosis and treatment of strep throat could be more comprehensive in the future. As far as the genetic pathway goes, it's a little bit more difficult and bioethically controversial. A promising new technology, as mentioned before, CRISPR-Cas9, could be utilized to repair these genetic mutations in GJA1. And at the beginning of February, British Parliament actually legalized this process for initial testing on human embryos, which proves that this technology isn't as far off as it may seem.
So we'd like to thank our supervisor, Dr. Kim Campbell, for her excellent help in helping us do this project. And now we would like to open ourselves to your questions.
So for the strep side of it, is that just if they've ever had strep before, or if they currently have it while the fetus is being developed?
So what we are proposing is that the antibodies produced while someone has strep throat, and as we briefly touched on, there's a seasonal factor that actually another person's project, the nitrous oxide project, was also focusing on the seasonal aspect, in which there's an increase in summer and decrease in winter. Now, if we shift that by 6 months, we see that there's an increase in people receiving HLHS developing in late fall, winter, and early spring, which is when strep throat increases in prevalence. So that what we are suggesting is that it is strep throat being the reaction to strep throat during the pregnancy.
Okay.
Have you looked at possible other infections that obviously would use antibodies as well as being a cause for HLHS, or is it just that you've read about strep throat, or?
Right, so that's a good question. Is well, first of all, why isn't it that more of these IgG antibodies would affect this gap junction? But what we found in the research is that there's a correlation between these IgG antibodies and hypoplastic left heart syndrome in a rat study. But what we were really the reason behind that is because each antibody is specifically formulated to respond to a specific antigen, and there are very minute differences between the different IgG or between the different antibodies. The sequence of amino acids, they're just very slightly different. And what we're saying here is that that is so specific, and also that that phosphorylation tail is also very specific, and it just happens to, and we have data that supports that they happen to interfere with each other.
Regarding the gap junctions, could that just be specific to HLHS, and in what way is it specific, or could it encompass other congenital heart defects?
Well, we found that connexin 43 is most widely expressed in the heart, and it exists around the body, but it's most widely expressed in the heart. And so impairment of the connexin 43 would mainly affect things that are in the heart. We didn't, I guess, look for this with relation to other heart defects. I don't know if we found much about that, but right, it's most widely expressed in the heart. So I mean, that would explain the variety of different traits that are observed in HLHS.
Can you address the left side of the heart being more affected by gap junctions than these malformed gap junctions than the right side? Like, how is this present specifically in HLHS?
So on the environmental aspect side, we had looked at, in our study, they had explained why in all the rats only left-sided malformations occurred, and they did this. They explain this because of the tendency or the preference to go from the placenta into the left side and the right side. Not saying it only will go that way, but they tended to do that more.
It's basically, it cycles in the left side more so than in the right side, so there's a higher chance of the antibodies to interfere with tissue in the left side.
Higher concentration of antibodies.
Exactly.
So is there a disruption, going off of that, is there a disruption in the coronary arteries then, as to how it's reaching the myocardial tissue?
Well, we're with the openings that are between the left and the right side of the heart, mainly the oval. Yeah, the oval. That allows the blood to circulate straight into the left side, which is only present in the developing fetus. I think that your diagram was actually of a heart that's post-birth, and it didn't have those holes in it, and so I think that's where the miscommunication is.
But wouldn't most of the exchange come through capillary interactions, which would not be within the ventricles?
Can you say that question again?
Wouldn't most of the exchange rates, like if you have these antibodies present in your blood, you know, there's this question of surface area to volume ratio, and on the inside of your ventricles, there's certainly tissue present that's open to these, but once it leaves the ventricles, it's going to enter the coronary arteries and be expressed and be pushed throughout the entire myocardial tissue. And so wouldn't the, if it's in the entire blood, then wouldn't it have even distribution once it reaches that point?
Within the heart, are you saying, or in the whole body?
Within the heart.
We're just saying because the blood flow, it can, because you got the.
Yeah, I mean, the correlation was between these things and the left side of the heart. We're unable to, there's more research that needs to be done on this specific topic, but as we've said, we found that the data supports that these things correlate with the left side of the heart.
They might be pushing you on that because I was pushing them at their poster on this.
Right.
I want to be clear though, is there a direct link between the antibodies and connexin 43? Is there evidence or citation that those antibodies actually bind to that protein?
With the connexin 43?
Yeah.
Yeah.
The say that.
So the antibodies, the maternal antibodies they're actually able to bind to the connexin 43 protein?
We know that the anti-strep antibodies, when they were injected into the heart, that they produce the HLHS phenotype. On a separate route, we know that the class of IgG antibodies can block the phosphorylation sites on connexin 43 carbonyl tail.
Okay.
So it's
The left side and the right side of this diagram indicate causative structures, whereas our correlation and our ultimate hypothesis is at the very bottom.
Sure. All right, very good, thank you.
Good afternoon. My name is Kaylee Evers from the University of Minnesota, Rochester, and my project today is putting the brakes on hypoplastic left heart syndrome. If we start with a little bit of background, this condition was first noticed by Lev in 1952 when he saw the underdevelopment of the aortic tract. This was expanded by Noonan and Nadas in 1958 when they saw that the left atrium and ventricle was also affected. At this point, it was retermed hypoplastic left heart syndrome. If we compare the hypoplastic heart to the normal heart on the left side here, we see that the left atrium, the mitral valve, the left ventricle, and the aorta are all severely underdeveloped. In addition, we have the ductus arteriosus and an atrial septal defect. So why is this important? If we look at the prevalence of HLHS in relation to other congenital heart defects, it's relatively small, only 7% of cases. If we look at the mortality associated with this, it is one of the highest. In fact, it is the leading cause of death among congenital heart defects. And currently, there is not a cure for this condition. We only have 2 treatment options, including a complete cardiac transplant or a series of 3 surgeries, all before the age of 2. Currently, we do not know the cause of this condition either, though current studies have looked into genetic or environmental causes. I decided to delve deeper into the environmental causes when I saw that most of them focused on fine particulate matter. There was not any research done on actual sizable molecules. For this reason, I have hypothesized that hypoplastic left heart syndrome is caused by nitrogen dioxide in the mother's environment. If we look at this graph here, we have the prevalence of HLHS adjusted for when it begins developing around the 10th week. And we see that it peaks in November, December, and January. If we compare this to the seasonal rises in the nitrogen dioxide levels, they also peak in November, December, and January. So I've devised this schematic. We have all the cars on our roads today emitting a massive amount of nitrogen dioxide. This nitrogen dioxide is inhaled by the mother where it enters her respiratory tract. After the gas exchange in the lung, it enters her cardiovascular system. This is then connected to the fetal cardiovascular system, where blood flows from the umbilical cord into the right atrium. So once this nitrogen dioxide is inside of the fetus, we have two effects: hypoxia and oxidative stress. With hypoxia, we have the presence of nitrogen dioxide lowering the partial pressure of oxygen, which is a necessary nutrient in order for the development of cardiomyocytes. So instead of having the growth of these, our cells are shrinking and dying. With the oxidative stress, we have nitrogen dioxide as an oxidant, causing an imbalance of oxidants and antioxidants. This imbalance disrupts the cell signals between cardiomyocytes, leading to an arrhythmia, which will therefore lead to cell death. So why does this only affect the left side of the heart? The arrhythmia would normally affect the entire heart, except in the fetal heart, the right atrium is semi-regulated by the flow of blood from the mother and is therefore not as affected by the arrhythmia. As you can imagine, the areas of the heart that work the hardest develop more, and the areas that do not have to work as hard do not develop as much. With the nutrients, we have blood coming into the right atrium, being very nutrient dense, and the right side of the heart and its structures receives all the necessary nutrients. From there, the blood can circulate to the rest of the body, and the body receives the nutrients it needs. When the blood returns to the left side of the heart, we see little to no nutrients left. In summary, we have the shrinking and death of cells caused by hypoxia. In addition, cell signal blockage, arrhythmia, and cell death due to the oxidative stress. And we only see the left side of the heart affected. In order to test this, I would grow populations of cardiomyocytes in the lab and expose them to varying concentrations of nitrogen dioxide to see which concentration was needed to yield these effects, in which case the cells would be shrinking and dying. Once I have determined this concentration, I would look at the solubility of nitrogen dioxide in the blood. I would do this by bubbling the nitrogen dioxide into a test tube of blood, capping it, and letting an equilibrium be reached. After this, I would uncap it and measure the amount of nitrogen dioxide being released to calculate the amount that was actually soluble. So the significance and innovation of this hypothesis. This is a novel hypothesis, as the nitrogen dioxide as a cause has not been previously looked at, nor has the concept of nitrogen dioxide being soluble in the blood. Additionally, support of this hypothesis would lead us to define an at-risk population of women living in major cities near major roads and highways. Upon verification of this at-risk population, we could potentially prevent this terrible syndrome by removing them from that environment and giving them the oxygen they need. As part of this presentation, I would like to provide a huge thank you to Professor Haynes, Dr. Wright-Peterson, Professor Winship, and my faculty advisor, Dr. Terrell. Additionally, I would like to thank the Impact Program for providing me with this wonderful opportunity, and for all of you for your time and attention today. Any questions? Yes.
First, I'm really impressed that you did this by yourself.
Thank you.
Since obviously there's car exhaust a lot of places, what dictates, do you know, whether somebody develops hypoplastic left heart syndrome?
As with most environmental causes, some people are more susceptible than to others. Particularly women with lung disease or heart disease already could be increasingly affected by this. Unfortunately, we do not have a defined system for the surveillance of congenital heart defects to be able to link it to an exact population. I tried looking into other areas of the world like Beijing where there's a lot of pollution, but they don't have a system to keep track of these. In addition, if we are keeping track of these cases, they're kept track of by hospital, not by the woman's actual location. Yes.
Have you thought of like a curative therapy or something to counteract the effect of the nitric oxide? So if they were to be found to have this, is there a way to maybe reverse it, or?
Hopefully by delivering them oxygen, we could, they wouldn't be receiving any more of the nitrogen dioxide, that we could hopefully at least prevent it, we could actually stop the development of it further. But no, most of this hypothesis looks at preventing it altogether. Yes?
So you're proposing that this is the only pathway that leads to HLHS?
This is the only one that I have looked into specifically, but I am sure with a disease or a syndrome as complex as this one that there's likely other pathways as well. Yes?
Are there seasonal differences in nitrogen dioxide emissions?
Yes, there are. In the winter, I'll flip back to that graph. So if you look on the graph, the graph on the right side here shows the monthly levels of nitrogen dioxide, and there is a general peak in the winter months. Yes?
So are you suggesting that the underlying cause is the hypoxia?
The hypoxia caused by nitrogen dioxide. This nitrogen dioxide would lower the partial pressure of oxygen, resulting in the hypoxia. Yes?
Since NO2 is like really present in like a lot of communities, why do you think that it's showing up less as a congenital heart disease than other ones? Because in that graph you had, HLHS was like a really small occurrence, but then the other ones are really big. So even though NO2 is like really all over the place, how would like you explain the so much little HLHS occurrence with that?
It would have to do likely with the solubility in the blood, the amount that was actually able to reach the fetus. Although there is a lot of it surrounding it, it does not affect most of the normal population. Yes?
Do you have any information on HLHS occurrences and like by year and the amount of nitrogen dioxide present by year?
I don't have an exact figure, but in general there is a trend, yes.
And it's trending upward as we have more NO2 in the environment. It's
Yes.
up.
Sorry.
Questions? She went fast, so she had lots of time for questions. That was a dangerous thing. Dangerous time for questions. All right, thank you.
Good afternoon, everyone. My name is Ali.
My name is Opoku.
My name is Yui. My name is Nakumara. And my faculty mentor is Dr. Penny Novick, and we're students from Minnesota State University, Mayo. Today, we're going to be talking about hypoplastic left heart syndrome, HLHS, as a result of abnormal NEF2C activity and other related factors. To begin, what is HLHS? It is a congenital heart disease that affects about 1 in 4,344 infants in the U.S. annually. It is characterized by non-developed to small left ventricle, a smaller absent mitral valve, a smaller absent aortic valve, and sometimes a very narrow ascending portion of the aorta. To understand HLHS, it's quite important that we look at the stages of fetal heart development. This picture shows just that. So these are the stages of fetal heart development, and these are some important genes that play a role in development. The genes in blue, like TBX1, plays an important role in right ventricle formation. The genes in purple, like GATA4 and NKX2.5, play an important role in left atrium and right atrium development. And finally, the gene in red, TBX5, plays an important role in left ventricle formation. It is very important to take note that all these genes are highly interconnected, and a downregulation of one gene might affect another.
So with this in mind, we started off by wanting to know the expression levels of these major genes, which, as he just mentioned, are important in the left-hand side formation. So what we found in a review paper where they used induced pluripotent stem cells and measured mRNA transcript levels was a decrease in HAND1, HAND2, TBX2, and NKX2.5. In addition to that, there was a decrease in differentiation potential of cardiomyocytes. Thirdly, there was a decrease in Notch and Hay signaling in these. An important note about Notch is, in another paper, we found there's a strong correlation with mutations in the Notch signaling pathway and the appearance of HS. So with that in mind, having a signaling pathway and all these transcription factors, we kind of wanted to find something in the middle that can be affected by Notch and also affects the expression of all these, which was the myocyte enhancer factor 2C, which, as I said, plays a vital role in gene expression. So our hypothesis is, during critical periods of heart development, abnormal activity of the myocyte enhancer factor 2C as a result of irregular Notch signaling and folic acid levels, as well as genetic mutations, is the underlying cause of HL-HS. When looking at Notch, what we noted was Notch actually inhibits the expression of MEF2C. So when you have high Notch signaling, MEF2C goes down. When Notch signaling is low, overexpression of MEF2C occurs. Why is MEF2C so important? In addition to what I mentioned on the previous slide, MEF2C also works with MMP2, or the metallinose proteinase 2, which is important in cytoskeletal remodeling. It also ensures that the left side of the heart grows faster than the right side of the heart. This is just a schematic showing MEF2C, and it's important in binding and activating cardiac muscle gene proliferation. As I mentioned earlier, overexpression of MEF2C affects GATA, which on the diagram we showed earlier is important in left-hand side formation. GATA also alters cushion formation and remodeling in the heart. These are just a few of the transcription factors that we mentioned, just to kind of show you once again, localized to the development of the left-hand side of the heart. So that's why with these factors being mutated or not functioning properly, it can explain many of the phenotypes that we do see.
As my colleague mentioned earlier, that Notch signaling affects MEF2C. Each levels of Notch signaling could affect how MEF2C is being regulated. Notch1 receptor has been strongly linked to HLHS, so the main Notch1 receptor we're looking at is the main receptor we're looking at is Notch1, which mutations of this gene could cause HLHS. So this is just a schematic showing the reaction with MEF2C and Notch1, how they could affect. And this shows MEF2 dependent receptor. The presence of MEF2C on lane 3 shows you an increase in full activation of this receptor, while the presence of Notch1 on lane 4 shows how the full activation decreased, including if the presence of MEF2C is there. Also, this is just a schematic showing how Notch inhibits MEF2C. So if Notch signaling is reduced, overexpression of MEF2C is increased, which will affect GATA and KX2-5 and TBX5 that affects the left side of the heart. And also we went on further by looking at what factors could affect Notch signaling, including MEF2C. Folic acid has been shown or be strongly linked to increase Notch signaling. So here on this diagram, the control shows a normal level of folate, and folate L shows a less increased level of folate, and Notch signaling increased as folate increased. Folate H shows a high increased level of folate, and Notch signaling increases for. Furthermore, we looked at different review articles and literatures that shows that talked about folic acid and some other folic acid and not signaling. Now we found out that folic acid inhibits HLHS. In Baltimore, HLHS have been shown to be prevalent, and industries is high in the city, which industries eliminate industrial solvents such as trichloroethylene. And trichloroethylene in a different article has been shown to cause folate deficiencies in mammals. In this diagram, a study done with mammals on how trichloroethylene could cause folate deficiency. As trichloroethylene was increased, folate deficiency increased due to presence of presence of folic acid being released from the urinary sample, and the mammals are unable to take in the folate.
So to summarize all we have talked about, decreased folic acid as a result of a chemical, as Kasim talked about, will lead to decreased notch signaling through the notch-1 receptor, which in turn will lead to an increased level of MEF2C, because they're inversely related, and that will lead to that would affect some transcription factors and genes like TBX1, GATA4, that are necessary for myogenesis and differentiation in the heart, which we believe is a cause of HLHS.
So now I'm going to go over the key points, what we plan to achieve, and how we plan to test our hypothesis. So our hypothesis focuses primarily on the combined genetic and maternal effects causing HLHS. With respect to genetics, we're looking at the MEF2C gene and the NOTCH1 signaling gene and their interaction between each other. And the maternal effects is focusing on folic acid levels and how the presence of the folic acid in different concentrations can cause the onset of HLHS. What we plan to achieve with our hypothesis is we plan to change the diagnostics of HLHS. Currently, HLHS is diagnosed via ultrasound during week 14 of gestation, or it's diagnosed after birth, where they just look at the baby's physical properties after birth, such as observing a cyanosis, which could be indicative of HLHS. With our hypothesis, we plan to have embryonic genetic testing done very early on in the pregnancy so we can identify, if we identify these genes, MEF2C and the NOTCH1 signaling genes, as factors causing HLHS, if we observe that there are decreased expression or decreased DNA sequences of MEF2C and NOTCH genes in an embryonic genetic test, then we can identify them as causation of HLHS and start working towards treating it much more quicker. So for testing our hypothesis, we plan to use a gene knockout analysis. What we'll be analyzing is iPS stem cells, which we will differentiate into mini heart cells, which is shown a diagram right here. The idea for this study came from a lab in California that actually was successful in differentiating iPS stem cells into small mini hearts. The mini heart mimics the actual human heart and will allow us to identify factors that can allow us to identify the phenotype of HLHS. What we will use for the gene knockout analysis is a CRISPR-Cas9, which is really invaluable to our study because of the specificity of the cuts that it makes in the DNA sequences. Other techniques might cut too much or too little of the DNA sequence we want, and we want to be sure that it's actually the MEF2C and or other gene causing HLHS. We will also look at maternal effects on gene expression, which is again going to the folic acid levels and how that may play a role in HLHS. And the knockouts and changes in folic acid will be done around critical periods of heart development, such as day 21 through about 30, where the heart ventricles start folding and looping. We would like to acknowledge that the Biological Science Department of Minnesota State University, Mankato, our research mentor, Dr. Penny Knoblich, who was not able to attend, our director, Catherine Campbell, for helping sponsor this event, and Mayo Clinic for giving us the opportunity to let you guys hear our ideas. These are our references, and does anybody have any questions?
So that Baltimore study, I think we both referenced the same one from
Yeah.
the 80s. So in that study, I believe they did a synthesis of what they did an analysis of industrial compounds, and I believe that they found no correlation between the industrial compounds and HLHS. I guess my question is, wouldn't industrial compounds be more linked with HLHS in urban areas across the world rather than just Baltimore?
First of all, yes, I agree that that wasn't linked to HLHS, but that was linked to folic acid levels from a different paper.
Through the industrial compound,
Yeah.
though, right?
Yeah.
And so I mean so I think the study is saying is that it wasn't like an anomaly in the 80s with an extra amount of this industrial compound, that was consistent through more time than just that decade.
Well, given the prevalence of HLS in that area, we thought it'd be a good idea to just, it's an industrial area, look at different organic solvents that are being released in the area. And, I'm not aware that they said there was no correlation, but trichloroethylene being one of the things released, we thought it would be a good idea to look how it possibly affects folic acid. I'm not sure if in that paper they did look at that. How would you know at all?
I'm pretty sure they addressed it, but I might be mistaken. So.
Yeah.
Do you know what industry that compound is associated with specifically, or?
I do not. I just know it wasn't a lot of the solvent.
Is it used in dry cleaning?
Yep.
Yeah. So then I would wonder whether women who are working at dry cleaners have a higher incidence of infants that have this syndrome.
That's a good correlation, actually. What the study was actually looking at was just how folic acid is going to be changed, like the absorption is going to have through trichloroethylene. So there probably might be a decrease in folic acid absorption in somebody who's working in a laundry factory, because then, and the decrease in folic acid doesn't necessarily have to be HLHS, you get it right away. So if the folic acid levels decrease, it's something we found will change the expression of the MEF2C, and once that expression changes, then you have transcription factors like GATA4, TBX, and KX2.5 that are going to be affected. And since there are so many transcription factors that are being affected from folic acid levels changing, which changes Notch, and which changes, let me go back to the slide, which changes MEF2C levels, and then the transcription factors, that's why there's so many different phenotypes for HLHS. Because a lot of presentations have, I think there was one that covered the aortic valve, one of the gene that's involved in that one, I don't know who did the presentation, but somebody talked about aortic valve being changed by one of the transcription factors we covered is affected by MEF2C. So that's that question.
And to add to what he said, one of our major focuses is timing, so critical periods of time. So it will be really, really difficult to say people who work in dry cleaning places will have more, be prone to it. If it doesn't happen at that time, then you can't have it tell.
Question? Yeah.
As far as testing it or the transcription factors is concerned, would you be looking at the regulation of MEF2C, or would you be looking at the expression of the transcription factors in the genes to kind of analyze what's going on in that when you're talking about the tiny parts and that?
So you're talking about, are we going to be looking at MEF2C's expression or the transcription?
Yeah, so we'd be analyzing whether there's these transcription factors, because that's the actual cause and effect, or you'd be looking at the MEF2C for your...
I think we'll kind of do a little bit of both. It'll be kind of a good idea to look at MEF2C, but also see, obviously, what genes it is affecting. So a little bit of both would be probably.
And we would focus on MEF2C because we've identified it as something that's causative of affecting other transcription factors. So that would be the first primary place of our analysis.
Very good. Thank you.
Good afternoon, my name is Sol Jin, and this is Ryan Hershey, and we're representing Crown College. As I present, please ignore the font formatting. I think there were some technical issues with that, so I'll begin. Hypoplastic left heart syndrome is a congenital disease where infants are born with underdeveloped left heart. This causes an insufficient consumption of oxygen throughout their body. This picture over here is Camilo Lozano. Camilo's parents are very active in bringing awareness to HLHS. And just like Camilo, infants born with HLHS are required to go through open heart surgeries, just like Camilo did. But by finding the underlying cause of HLHS and with the application of regenerative medicine, we may be able to prevent this disease before the baby is born, and also give alternative methods to avoid open heart surgeries on infants.
So we looked at the two general factors that cause HLHS. First, we'll look at the seasonal factors here. This diagram was taken from a study done by Dr. Ettistetti there on congenital heart diseases, and it shows a trend of infants born with HLS during the summer months. This could possibly be because during the winter months, people are more prone to cardiovascular and autoimmune diseases, and when a woman is pregnant during that time, this could in turn affect the unborn fetus. So second, there are genetic factors to the disease. Dr. Hinton, in a study, stated that HLHS is highly heritable, which suggests there are the genetic factors behind HLHS. Our research focused on these genetic factors, especially in the mitochondrial functions. Mitochondrial functions are crucial because cardiomyocytes are dependent on the mitochondria, and the heart not only contains the highest density of mitochondria in the body, but is also highly dependent on ATP synthesis from oxidative metabolism. Cardiomyocyte differentiation is when embryonic stem cells are differentiated into heart cells. According to Folmes and other researchers, MPTP may be responsible for cardiomyocyte differentiation. MPTP stands for the mitochondrial permeability transition pore, and it's located on the inner mitochondrial membrane. This diagram here was taken from Congenia. It's a pharmaceutical company that focuses on the development of small molecule drugs that target the MPTP. Based on Folmes' study, the MPTP, the open MPT opening here, alters the mitochondrial membrane potential and increases reactive oxygen species inside the cell, also known as ROS. The ROS are reactive molecules responsible for the oxidation in cells, and too much oxidation in the cells can cause the oxidative stress, which can lead to a diseased cell and or cell death. Also, ROS was also found to delay cardiac differentiation, which in turn would alter full organ development. There are speculations that genes responsible for receptors that regulate MPTP cause the abnormal opening of these pores. These genes need to be understood, as it may be the cause of excessive ROS production inside the cell. While abnormal openings of the MPTP seem to be a promising cause of HLHS, we hypothesize that other mitochondrial genes need to be considered, such as FAHD1.
So the FAHD1 is located in the mitochondria, and it is a eukaryotic protein for oxaloacetate decarboxylase. As you can see in this figure, that means that the oxaloacetate are converted into pyruvate with the help of FAHD1 by releasing carbon dioxide. This shows that FAHD1 is responsible for the level of oxaloacetate and the pyruvate in our cells. In fact, FAHD1 deletion in mice was found to increase oxaloacetate levels, studied by Mayo. Observed during cardioprotective ischemic, which is the occurrence of reduced blood flow in the tissues, oxaloacetate was found as one of the inhibitors of complex 2. Oxaloacetate inhibits complex 2, as you can see here, and it decreases ROS production in complex 1, but it increases ROS production in complex 3. And if the increase of ROS in complex 3 is significant enough, this could delay cardiac differentiation and become a putative cause to HLHS. An interesting study was that complex 2 was found to be the primary entry point for the electron transport chain at embryonic day 9.5. This indicates the importance of the complex 2 in functioning the mitochondria and functioning the mitochondria during embryonic development. Therefore, if there is a FAHD deletion, there will be an increase in the oxaloacetate, and there will be more inhibition of complex 2. And I hypothesize that there will be more production of ROS and maybe delay cardiac differentiation and become a putative cause to HLHS. We propose 4 reasons FAHS1 needs further research. The bar chart here displays FAHD1 gene expressions in different organs, the red indicating the heart. Our first reason FAHD1 needs to be studied is because however low the expression is, it is still expressed. And all genes in the mitochondrial regulation at an embryonic level needs to be considered. Our second reason is because FAHD1 genes found in C. elegans, which is a homologue of human FAHD1, was found to maintain mitochondrial function and physical activities. It was also found to reduce oxygen consumption by affecting the pharyngeal pump, and the pharyngeal pump is generally known to be the model for organ development, which may be considered a model for heart development as well. So, according to Teffner, he did not see any significant problem of FAHD1 deletion in mice. However, our third reason to study FAHD1 is because there may be other factors that compensate for FAHD1. In contrast to our third point, however, FAHD1 deletion in mice increases oxaloacetate level found in the kidney and the liver cells. This leads to our fourth reason, because FAHD1 deletion in the heart cells are not fully understood. So the similarities between the abnormal MPTP opening and the FAHD1 deletion is that it alters mitochondrial membrane potential and influences ROS production. Suski stated that lower membrane potential and decreased activity of respiratory chain is observed with a simultaneous increase in ROS production. Regarding Teffner and Fohn's statement that FAHD1 deletion and the abnormal MPTP opening decreases mitochondrial membrane potential, we hypothesize that this will increase the ROS production and putatively cause HLHS.
So here we drew a model that depicts our hypothesis. The winter environment here and or the gene mutation causes a gene expression or repression that can either cause the abnormal MPT pore openings or FAHD1 deletion. These will ultimately affect the ROS production and cause mitochondrial dysfunction, which will compromise the heart development. Hence our hypothesis states that genes that increase ROS production involved in mitochondrial dysfunction in heart muscle cells, such as the proteins of the MPTP gateway and FAHD1, cause HLHS by compromising heart development.
So we connected our innovation to regenerative medicine. According to Holm, it stated that antioxidant treatment actually increases myocyte differentiation, and so rejuvenation of antioxidants can be used to control ROS levels in the fetal heart. Secondly, cardiac stem cells can be grown in an in vivo environment in order to study the regulation of MPTP mechanism and also study the effect of FAHD1 deletion and its effect in human heart cells. And thirdly and lastly, if FAHD1 deletion or the abnormal opening of MPTP is the underlying cause of HLHS, regenerative medicines can be used through the application of genetically modified cardiac stem cells on the fetal heart. Thank you.
So did you have a link to the winter environment, which incorporates it with the environment of winter to your hypothesis, or are you just suggesting that there is one?
Yeah, environmental factor. Mm-hmm. Yeah.
So at the end of your presentation, you noted that antioxidants might be able to help prevent HLHS. When would you recommend women, or particularly pregnant women, to take antioxidants, or just is any time fine?
Well, I think antioxidant consumption needs to be a regular part of a pregnant woman as their dietary regulation. But regarding the HLHS, I would think that when it's found that because if the MPTP opening is the ultimate problem, then it's said in an article that MPTP closes at embryonic day 13.5. So the problem comes when they're open after that time. So we would think that maybe consumption of antioxidants after embryonic day 13.5 could help. So.
It was my understanding you referenced a study that said that HLHS was very congenital. I think there was some contention about that earlier, and like talk about how HLHS is not like shown between very strongly shown between siblings. I'd just like a response to that. Like, how would respond to my mutations being the cause of HLHS when there seems to be some confusion about how?
Try to dig up the debate. Phone a friend if you need to. No, Dr. Olson.
So could you restate your question?
So mitochondrial, again, this is just a number. Mitochondrial DNA is supposed to be very conserved from mother to child.
Okay.
And earlier today there was talk about how HLHS is not like very strongly shown to be in two children at once.
Okay.
So how would you respond to that being with it seems mitochondrial coding being the source for HLHS in your and someone please tell me if I'm completely off on this.
No, I'm just not understanding it completely.
Dr. Olson, you have an opinion on that?
I'm not sure it addresses your exact question, but the lack of a family history doesn't negate the possibility of genetically disease. So one possibility is de novo mutations, and that's actually been a major area of focus in different research labs with heart disease, looking for mutations that have arisen spontaneously. So in that case, you won't have a family history. And the reason that wouldn't be transmitted then to offspring is because many people who have hypoplastic left heart syndrome will not go on to their children. Also, recessive inheritance is another possibility, in which case you could have two clinically silent genes coming from either parent, which conspire to cause hypoplastic left syndrome. And again, you might have a sibling that also has recessive disease, but again, another possibility to explain genetic underpinnings that aren't necessarily in the context of
Have you ever heard of the concept of heteroplasmy? No? Has anybody ever, and Esther, you can't answer this. Anybody ever heard of the concept of mitochondrial heteroplasmy? Ring a bell to anybody? Esther, can you explain it in 30 seconds?
Yeah, so the mitochondria has its own mitochondrial DNA, but there is not all the copies, they are not the same. So that means that they are different. That means heteroplasmic. So, and the disease starts to develop when the mutation gets a certain threshold in the heteroplasmic.
So you can have a combination of diseased and healthy mitochondrial mutation within the same cell, within the same individual, and depending on which tissue or cell can be healthy or normal. If you really are interested, Esther has a paper under review that actually shows this. And so I think your hypothesis here is really interesting in that context because there could explain some of this variable penetrance that we struggle to describe when we have nuclear genomics as our primary explanation. We put mitochondrial genomics on top of it, and boy, it becomes exponentially more complicated. So I think this is a very relevant topic. So good job. Thank you. So I don't know about you all, but I think I know quite a bit about HLHS, and I learned some things today. I took a lot of notes, and there's some literature that I'm going to back and review. I'm not going to point them out right now to give any bias towards what my thinking is right now, but there was definitely some interesting associations that I haven't thought of, that our team hasn't thought of, that challenges us to think differently. So as much as this may seem like an exercise for you to go through the process, to put the talks together, to interact with each other today and to learn, I think I'm quite confident it was an extremely valuable experience for you, but what you might not fully appreciate is how valuable of an experience it is for us that are here at the program. And Dr. Olson and the postdocs and senior scientists in the program are here listening to you. So we appreciate the energy, the effort, and the commitment that all of you have put into this. And it really is quite remarkable. And I'm really have a lot of gratitude to be part of your symposium today. And a big shout out to Katie and everything that Katie has put together to make this happen. So Katie, any final words before we depart? What's our next step here?
The next step is lab tours. So if after all of these presentations you still remember your group number that you got when you walked in the door, you'll find your escort right outside these doors, and then you'll go on, each group will cover all 4 tours, but you'll start with your group number.
Thank you.
Thank you.